Good afternoon viewers and welcome to the EduSat lecture for the science stream. Today we have with us Dr. Charu Dogra Rawat, who is an assistant professor at the Ramjas College, University of Delhi. We have been having Dr. Dogra in our EduSat studios for various lectures on molecular biology, and continuing that stream we will be talking about DNA topology. We have in our previous lectures understood that DNA is a double-stranded molecule. But what does this double strand exist like in the cell? Is it always linear, or like any rope kept in a glass of water would undergo lot of turnings and lot of knots? So is there a way of characterizing these knots and turns? Do they have some functional significance? And how can we characterize different DNAs from different organisms on the basis of these knots and turns, which broadly is the term topology. Also, we will talk about another property of DNA, which is DNA melting. DNA as a molecule is very resilient. You heat it, it again coils back to its double-stranded structure. So we will be looking at DNA denaturation and renaturation and Cott curves. So over to Dr. Charu Dogra Rawat.
Thank you, Uniti. As she rightly put it, I am going to talk about three distinct topics today, which is DNA topology, DNA melting, and probably organelle DNA. Now, as she said that we have studied about the structure of DNA, it's two polynucleotide chains which are coiled around each other in a helical form, and but that's how we theoretically know it. How does it exist in the cell? Is it just like a thread which is there? So we'll talk about that the existence in the cell is more like in the form of various kind of coils. So DNA topology is basically, so the learning objectives for today's talk will be to understand the linking number and its components of a covalently closed circular DNA molecule. After this lecture, you will have an understanding of how cells utilize this supercoiling. You will have the understanding of the DNA melting, which is basically dissociation of double strands of DNA when an external factor is applied, its reassociation and its relation to the complexity of DNA. You will have the understanding of the features of extranuclear, which is primarily the organelle DNA. So DNA topology will start with. DNA topology is described as the coiling of the molecule of DNA. So how is this DNA molecule coiled inside the cell? Now, most of the eukaryotic chromosomes are linear, and these linear double helical structures are freely able to rotate and unwind so that the coiling of the two strands with each other, the number can be changed. So because the two ends are free of a linear DNA molecule, so the two strands can rotate and unwind in a manner to change the number of coils which is existing between them. But there are also circular DNA molecules which are present, such as in the case of bacterial chromosomes. Most of the bacterial chromosomes are circular in nature, some viral chromosomes are circular in nature, and all these circular molecules and some extra chromosomal DNA molecules, which are termed as plasmids, are also circular in nature. So the circular DNA molecules, the two strands are covalently closed circular DNA molecules, and their actual number of the times the chain's coil cannot change. So inside the cell, these are just the models which is represented here. This is now how the exact DNA looks like in the cell. But this is just to explain, these models are shown here to understand it better. So the covalently closed circular DNA molecules, because the two strands are covalently linked together, the actual number of times the chain's coil cannot change. So these DNA molecules are said to be topologically constrained. So topological constraint factor can also come up in the linear DNA molecules when, for example, they are associated with certain protein molecules. So generally linear DNA molecules can freely rotate and unwind, but when they are associated with certain protein molecules, they also come under a topologically constrained condition, as shown in the diagram. These molecules basically if you look at, if you want to separate these two strands, then the strands cannot be separated until and unless a covalent bond is broken and one of the circle is opened. These two strands, you cannot, how much do you distort the molecule? You cannot separate these two strands until and unless you cause a break in the covalent bond and one of the strands open the circles. That's the only possible thing, and that is why it is said to be topologically constrained, because these twists which is there, the coiling, it cannot change, and to separate these two strand molecules, one of the strand has to be broken and then only these two can separate out. So, or somehow if you assume it or if you think it in a manner that some kind of a magic occurs and at the points written as 1, 2, 3, 4 and 5, somehow the two strands are allowed to pass over one another, then only is the possibility that the two circles can separate out. So, the two strands which are passing over one another at these numbers 1, 2, 3, 4 and 5, somehow they have to come out and then only the two strands can separate. So, what we define this number 5 is the linking number. So, the amount, the number of times the two strands have to cross one another to become completely separate is defined as the linking number. Now, linking number is always an integer. It represents the number of times one strand of DNA would have to be passed through the other strand in order for the two strands to be entirely separated from each other. Just like we saw that if some kind of a magic happens and all these at these points where the two strands are crossing one another, they just somehow comes out. So, that is defined by the number LK, which is equal to 5 in this present model. This linking number is an invariant topological property of CCC, covalently circular closed DNA. No matter how much the cceDNA is distorted, the LK remains constant. As I just told you that the two strands cannot separate until and unless you break open one of the circles. So, until and unless there is a breakage of the covalent bond, the two strands cannot separate and the linking number cannot change. So, linking number is an invariant topological property of a closed circular, covalently closed circular DNA molecule. Now, this linking number is a geometrical progression of two numbers. One is called as the twist and another is called as the writhe. So, we will look into a little deeper on how this linking number is achieved at. Twist number, if you look at, is the number of helical turns of one strand of DNA about the other. So, basically at how many points one of the strand crosses the other strand is defined as the twist number. So, the number of times one strand will pass over the other strand is defined as the twist number. So, if you look at in the figure, then their number 1, 2, 3, 4, 5 are the numbers where the strands are passing over one another, and therefore, that is the twist number. The twist number in case of a relaxed molecule of a DNA, the twist number will be equal to the linking number. So, the number of helical turns of one strand of DNA about the other. So, the model which we are considering right now has 5 turns, 5 twists, and therefore, the linking number is 5. Now, the most of the DNA molecules in normal conditions exist as a right-handed helix, and we did cover this in our DNA structure lecture. So, right-handed helix if you look at, then by conventionally the twist number is given a positive number. Right-handed helix you can identify by looking at the front strand which is basically moving up and towards the right, which is shown by a red arrow here. So, the strand on the front, if it moves up and right, then it is supposed to be a right-handed helix, and for a right-handed helix the number of twist is given as a positive value. So, if TW is greater than 0 for right-handed helices. If it is a left-handed helix, as it occurs in some conditions, for example, the zDNA we talked about, then by convention the twist number is giving a negative sign. So, twist number in this particular case will be -5. And how can you identify a left-handed helix? That the strand which is in the front is moving towards the up and the left side. So, if moved towards the up and the left side, then it is said to be a left-handed helix. It is not existing in the nature, but somehow due to certain physiological conditions, some part of a particular DNA or some DNA of a kind can exist in the left-handed manner. So, if we are considering the if the supercoiling of a left-handed helix will give the twist number a negative sign. So, if TW is less than 0, then it is a left-handed helices. Another structural component which we talk about is called as the writhe, because when it is lying in the cell, then the circular DNA molecule is also twisted on its own axis and multiple times. So, repeated times on its own axis, this relaxed DNA molecule can be twisted. So, it is this particular property is defined mathematically by a number called as the writhe. So, writhe is the torsional stress of a DNA molecule in which the DNA double helix crosses over itself in the 3-dimensional space. So, there are 2 strands of DNA, the 2 strands are crossing over one another forming a double helix, and then this helical structure is again coiling over itself on its axis in the 3-dimensional space to give rise to the coiling which we are talking about. So, this component, when it is twisting around its own helix, then it is defined as the writhe. So, if we look at the writhe portion, the component, then there are 2 forms of writhe which we talk about. One is called as the interwound writhe. So, interwound or plectonemic writhe that it is called as is writhe in which the DNA double helix wind around itself to form a helix. So, if it is coiling around its own axis, then it is as shown in the figure. If it is coiling around its own axis, then it is called as the interwound or plectonemic writhe. Then we talk about different kind of DNAs, like if right-handed DNA forms a right-handed interwound helix, then DNA must twist to accommodate the writhe. So, what we are talking about here is that 2 double strand DNA, when formed in the form of a double helix, how are they arranged inside the cell? So, obviously, if it is a circular molecule and some kind of a torsional stress develops, then when it twists on its own axis, then the DNA level twists have to compensate for it. So, if a right-handed DNA forms a right-handed interwound helix, then the DNA must twist to accommodate the writhe, as it is shown in the figure. So, if what we see is that the writhe in that case is less than 0 for right-handed interwound turns. So, we give it a negative value. So, writhe is given a negative value. So, we can see in the numerical written there that we were talking about the linking number, which is 5 in our model. Now, if you look at the twist, then if it becomes 6.3 and the writhe becomes 1.3, the important point to note here is that I told you that linking number is always an integer, but the twist and writhe can always have the fractional values also. But since the linking number is constant, the twist and the writhe, if the twist is increases, the writhe will decrease. If the writhe is increasing, then the twist will decrease. So, that means that if in a right-handed DNA, if you are introducing a right-handed interwound helix, then the twist have to accommodate that. So, the twist number will increase and therefore, the writhes will decrease. So, that is how it goes on. The twist and R, as I told you, can have the fractional values. Now, if the right-handed DNA is forming a left-handed interwound helix, so if you can see the first part of the diagram in both these cases is the same. It is the same 4 writhe it is showing, and that because we are just considering the backbone, we are not, as I told you, these are just the models to explain. This is not actually the structure of the DNA molecule. So, that backbone model is having 4 writhes, because the entire circular DNA molecule is twisted, but in the inset it is shown that when these 4 writhes are creating, then how the twists is accommodating that. And if the right-handed DNA helix in this case is forming a left-handed interwound helix, then left-handed when the front strand of the DNA is going towards left and up. So, left-handed helix when it is forming, then you can see in the inset that the twists are reducing. So, when the writhe is in the positive sense, then the twist is in the negative, and if the writhe is in the negative sense, then the twist is in the positive, because the LK, which is an addition of both these numbers, remains constant. So, LK remains constant, the twist and writhe, and therefore, we give them, assign them the signs because we were giving twist a positive number. So if it is doing it in the same manner, then we give the writhe as the negative number. So that's how it goes. So if you can see, then the writhe is greater than 0 for left-handed interwound turns. So linking number still remains 5, but because the twist is 2, because right-handed DNA is now forming a left-handed helices, then the writhe is increasing to be 3, so the twist is decreasing to form 2. So that's the whole concept of it, that the linking number of any cceDNA remains always constant, but the two components, the twist and writhe, changes. So twist changes when the number of times the two strands are interwound onto each other changes, and the writhe changes when this entire DNA molecule, either on its axis or in association with the other protein, coils together. So that's the whole concept of it. The another kind of writhe which I am talking about is also called as the spiral writhe, and spiral or toroidal writhe is the writhe in which the DNA double helix winds in a cylindrical manner, for example say on a protein molecule. So one thing is interwound writhe, in which it is making a helix onto itself, onto its own axis, but there is another possibility that it can wound around a protein that occurs in the normal chromosome. We will be talking about genomic organization down the lane, but if you see, then if you know about it, then DNA in the genome is actually a DNA and a protein molecule. So the proteins are the histones primarily and some non-histone proteins, but histone proteins, and then the DNA is wound around it. So when the DNA is winding around a particular molecule or protein molecule, then that kind of a winding is called as the spiral writhe or toroidal writhe. Now if right-handed DNA writhes to form a right-handed spiral coil, then the twist has to decrease. So this is basically opposite of the interwound helix. So what was happening in the interwound helix, that when the interwound writhe was occurring, then if right-handed was doing in the right-handed direction, then the twist was increasing. If a right-handed was doing in the left-handed direction, then the twist was decreasing, because twist was going to accommodate the number of writhes which were occurring. But in the case of spiral writhe, because when a right-handed DNA writes to form a right-handed DNA helix, the TW decreases. And I told you that linking number has to remain constant. So linking number has to remain constant, so when TW decreases, WR increases. So for this cases, the WR will be given a positive sign. So LK is still 5, but the TW is equal to when it is decreasing, and as I told again, it can be fractional values. So writhe in this case will be positive. For spiral writhes, it will be opposite of what was in the interwound turn. So WR is greater than 0 for right-handed interwound turns. If right-handed DNA now writes to form a left-handed spiral coil. So we are talking about two different type of writhes. We are talking about two different handed DNAs, the same right-handed DNA, which is the most common kind of DNA which is found in the cell, and then we are trying to coil it either in the right-hand direction or in the left-hand direction. And then there are two different things which we talk about. One is when it rotates around its own axis, then what happens? And one is when it rotates around a particular protein molecule or a cylindrical whatever, a component, when it is rotating around, then what happens? So broadly speaking, the two things are opposite to each other. When it is rotating around its own helix, then if the right is rotating in a right manner, then it is basically given a particular kind of a value. So if right-handed DNA writes to form a left-handed spiral coil, DNA must twist to accommodate the writhe. So the writhe in this case will be negative because the twists are increasing. So again, just to repeat this thing again, because it's a little confusing, but you have to remember this, that in the case of interwound writhe, if it is a right-handed interwound turn, then the writhe is less than 0, it's given a negative value, and writhe is greater than 0 for left-handed interwound turns. And mind that this is all we are talking about is the right-handed helices. So right-handed helices doing a right-handed interwound turn, writhe is less than 0. Right-handed helices doing a left-handed interwound turn, writhe is given as a greater than 0 value. And the same is opposite for the case of the spiral writhe. And the difference between interwound and spiral, that interwound is doing on its own axis and spiral is doing around a protein molecule. So I hope you understand it in a little clearer manner. Now we come to talk about the linking number of the relaxed DNA. So we know now that the linking number, there are two components. We will talk about that when a DNA molecule exists in a cell, then first we start with the default situation, that we talk about, okay, let's see that if the DNA is just in a relaxed circular DNA molecule, then what happens? We have already known about the structure of DNA, and we know from one turn to another turn there are around 10.5 base pairs of DNA which are there. This is a B form of DNA. We talked about the different forms of DNA. So, the most commonly existing B form of DNA, one helical turn is equal to 10.5 base pairs of DNA. So, we can easily calculate this linking number theoretically by finding out the base pairs of DNA divided by the 10.5, because if you remember, then I told you that linking number is the number of turns it makes. So, number of turns from one turn to another turn, there are 10.5 base pairs of DNA. So, if you know in a whole total DNA how many base pairs are there, for example, if there are 305 base pairs or 300 base pairs, then in a 300 base pair DNA, if you divide it by 10.5, then you will get a linking number of the relaxed DNA. So, linking number of the relaxed DNA you can calculate in this manner. For example, in this case, if there are 378 base pairs, then you can directly divide it by 10.5 and you can get that approximately there are 36 turns which are present in this. So, 36 times one strand will cross the other strand, and when one strand crosses the other strand, it is the linking number, because in this case if you can observe, then the writhe is 0. Because the DNA is in a relaxed state. So, LK is completely equal to TW and plus WR is 0. So, LK will be equal to 36. This LK of a relaxed DNA is defined as the LK 0. So, LK raised to the power of 0 is defined as this linking number of the relaxed DNA. So, now what is the linking number of the supercoiled DNA? We will talk about that, because as I was talking that in the cell, the DNA is not lying in a relaxed manner. It is under a torsional constraint. The torsional constraint in circular molecules is occurring because of the conditions which are present, and moreover we will talk about the genomic organization. So, genomes are large and they have to be compacted and put into a very small nucleus. So, if you have to package them into small nucleus, you have to coil them. They cannot be straight away, they cannot lie in a relaxed manner and fit inside that small nucleus. So, they have to be packaged in certain coils and knots and all kind of turns, and that is how they fit into a small nuclei. So, for the fitting into a small nucleus, the supercoiling has to be done. So, we are now defining this way to understand this supercoiling. We are defining it in terms of some mathematical numbers, and also like what this supercoiling is. How do we know that a DNA is supercoiled? This linking number of supercoiled DNA, if you look at this figure, then now in a relaxed DNA we have introduced writhes. So, the relaxed DNA is now on the left hand side, if you see, then there are 1, 2, 3 and 4 writhes which are introduced into it. So, this is a supercoiled DNA. It is a right handed interwound turns that have been introduced. Now, we know that the theoretical LK naught, which was of the relaxed DNA, was coming to be 36. Now, if you look at this number, the twists are 36, because in a relaxed DNA molecule the TW was 36, but because we have introduced writhes and recalling that a right handed helix is making a right handed interwound turn, so the writhe will take a negative value. So, 36 plus minus 4. So, that will give a linking number to be 32. So, for a relaxed DNA, the linking number was 36. But when you interwound it around its own axis, the linking number comes to be 32, because the other component writhe comes into the play. So, this is the linking number of the supercoiled DNA, is the difference between these two linking numbers. So, these are the two different states of a single DNA molecule. A single cceDNA, the linking number is constant in particular condition. If you try to change it, it will not change until and unless you introduce the component of the writhe. So, when you are introducing this coiling, then the linking number is changing. The LK naught was 36 and LK is 32. So, the difference between delta LK, the linking difference, the difference between the actual linking number and the theoretical calculated linking number, which is LK0, is defined as the supercoiling. That gives a measure of the supercoiling of the DNA, that how much the DNA is supercoiled. The DNA for which this delta LK is significantly different from 0. When will this difference be 0? When it is in the relaxed state. So, in the relaxed state, LK naught will be equal to the normal LK. If in a cell the DNA is existing in a relaxed state, then the relaxed state linking number and the actual linking number is the same. So, there is no supercoiling which has occurred. But if you see the actual linking number is different from the calculated linking number, then that difference gives us the idea of the supercoiling of the DNA. It has to be different from the 0. If it is less than 0, then it is said to be negatively supercoiled. If it is more than 0, then it is said to be a positively supercoiled. So, if you look at this circle, relaxed state circle of a DNA, you can twist it in one manner and you round its axis in a right-handed interwound helix, as well as you can twist it in the another manner in the left-handed interwound helix. So, if it is doing a right-handed interwound helix, we gave it a value of -4, because we talked about that right-handed DNA in a right-handed helix, and so linking number was becoming 32. For example, it twisted in the another manner on the other side. So, if it got twisted on the left hand side, then the writhe would have been positive. And if we are not allowing the change in the twist number, that is important, because we are not allowing it to accommodate the TW value. So, if it is not accommodating the TW value, and if the writhe is +4, then the linking number would have become 40. So, the linking number would have been increased, and now if you would have calculated delta LK, it would have been given a positive value. So, then the DNA is said to be a positively supercoiled DNA. So, if this delta LK comes out to be negative, then it is a negatively supercoiled DNA. If delta LK comes out to be positive, then it is be a positively supercoiled DNA. Now, cceDNA from both bacteria and eukaryotes are negatively supercoiled. So, this is the theoretical aspect of, okay, the DNA is in negative supercoil, it is in some kind of a torsional stress, but what is the significance of this torsional stress? Why this DNA is in this torsional stress? Why can't it be in the relaxed state? One thing is, obviously I told you that it has to be packaged inside the cell. So, for packaging inside, it has to accommodate a small volume, and for that you have to introduce the twists and turns and coils and knots and catenations and all kind of structures, so that this entire long DNA can fit inside the nucleus. That is one thing. But another important significance of this supercoiling is, which was described in 1965 by Wingrod's group, is basically that this negatively supercoiling is the store of the free energy, and this free energy that aids in the strand separation for processes such as DNA replication and transcription. So, what does that mean? If something is under stress, when that thing comes to a relaxed state, it will release energy, and this energy which it will release will be required for the double strand of the DNA to also dissociate. We will talk about that. Is why I said distinctly 3 topics, but I deliberately put them one after the other, because next will be how does this DNA separate. The 2 strands of the DNA separation, the DNA denaturation, require certain factors. You require energy to break the hydrogen bonds. If you remember, we talked about the structure of the DNA. The DNA, the 2 strands have a sugar phosphate backbone, and inside there are nitrogenous bases which are forming covalent, which are forming the hydrogen bonds. The A forms 2 bonds with T and G forms 3 bonds with C. If you want to separate these 2 strands, if you want to break the hydrogen bonds, you need to provide the energy. So, the replication occurs on a single strand. The transcription occurs on a single strand, because they read from a single strand DNA sequence. They do not read from the double strand sequences. So, the 2 strands have to be separated. So, for separating the 2 strands, you need the energy. So, for every process, the replication, transcription, you need the energy to be there, and that energy is stored in this supercoiling. So, when the DNA is present in the supercoiled state, and when the protein machinery comes in to either replicate it or transcribe it, then when it sits on the DNA molecule, then the DNA unwinds. And this unwinding will provide the energy for the 2 strands to get separate, so that the protein machinery can get the DNA single stranded template on which the replication or transcription, whatever the process is required can be done. So that is the main significance of this negatively supercoiling. That is how the cells are storing the DNA for, so that for these processes they can find out the free energy. So for any RNA polymerase or DNA polymerase, because it works on the single stranded molecule, the supercoiled molecule will become the relaxed molecule, and the energy which will be released will separate the two DNA strands also, so that the RNA polymerase or DNA polymerase can act upon it and either replicate one single strand or transcribe the strand to the RNA. When right-handed interwound turns in DNA relax, TW decreases and DNA winds, because it is the opposite. When initially we started studying when we were introducing. So in a right helix DNA, we were introducing the right-handed interwound helices. When we were introducing the right-handed interwound helices, we said that the twists increases. So it is obvious that it is the opposite case if we are doing, if we are in the right-handed helix, if we are relaxing the interwound turns, then the TW will decrease. So the twists will decrease, and twists are one strand over the other. So one strand over the other will decrease and the two strands will separate. So when right-handed interwound turns in DNA relax, the TW decreases and the DNA unwinds. So the linking number, the writhes become 0 and the linking number is equal to the twist number. Another ratio which we talk about is the delta LK divided by LK naught. So difference in this thing, which is indicator of the supercoiling, divided by the initial linking number, is defined by a term which is called as the sigma, and it is the superhelical density. So superhelical density becomes the intrinsic property of a DNA molecule, that what ability does it have to form the supercoils. So for that, delta LK by LK naught we can define it as. So there are other kinds of linkage also, such as catenation and knotting. So catenation and knotting, catenation is the interlocking of the DNA circles. So there are two, so we talked about the supercoils. If two different DNA molecules are associated with each other, then that is called as the catenation. So basically we talk about the supercoils, but there can be different structures which are present inside the DNA, that is how the DNA is present inside the cell. There can be catenanes, there can be knots, and the basically total linking is defined as the sum total of all these values. So we calculate the linking number to understand the supercoiling part. We understand the catenan part by defining a value such as C. We understand the knots part by defining a value such as K, and total coiling of the DNA molecule inside the nucleus is then defined by the sum total of all these numbers. So all these parameters are basically measured mathematically. There are equations. I just explained you about the supercoils, that how they are calculated, but similarly which are more complex as far as this level is concerned, and the catenans and knots, they can assign different numbers, mathematical numbers to it, and then they can find out the total linking of the DNA molecule inside the cell, and then comes the levels of packaging which is done. Now another thing is that if there is too much of a supercoiling in a DNA, if there is too much of a supercoiling in DNA, then the supercoils will tend to come to a relaxed state, because supercoiled is a torsional stress. So they will keep on coming to the relaxed state, releasing energy, separating the two strands even when the replication is not occurring, even when the transcription is not occurring. So if there are more, if there is no control on the supercoiling, and based upon the conditions, if it is kept in a large amount of supercoiling which is occurring, large amount of linking which is occurring, and then these linking can basically release a lot of energy which will unnecessarily create single strands, and then there will be a possibility of errors and mutations and damage to the DNA that can occur, because single stranded DNA is seen as not a normal thing by the cell machinery until and unless it is replicating or transcribing or something like that. So they have the cell has certain enzymes, and these enzymes control this supercoiling inside the cell. These enzymes are called as the topoisomerases. So now the topoisomerases, we have talked about two states of a DNA. One was a relaxed state and one is a supercoiled state. So it is the same DNA molecule of the same length, but in physiological conditions it can exist in two different forms. One is a relaxed state and another is a supercoiled state. These two states are termed as the DNA topoisomers. They are the same DNA, but their topology is different. One is relaxed and one is supercoiled. So, they are termed as DNA topoisomers, DNA molecules with the same sequence and length, but different linking numbers, because as we just saw that the linking number for a relaxed DNA and the supercoiled DNA was different. So, they are termed as the DNA topoisomers. And the interconversion between these two states is catalyzed by the enzymes called as the topoisomerases. So, topoisomerases are the key enzymes present inside the cell, which maintains that whether the DNA has to be in the relaxed state or whether the DNA has to be in a supercoiled state. The preferred state is obviously supercoiled state because it has to release energy, but I repeat myself again that the supercoiling cannot be too much, because that will be again harmful. So, to create a balance between the relaxed and the supercoiled state, the enzymes which play a role are the topoisomerases. So, we will talk a little more about the topoisomerases right now. There are two forms of topoisomerases if we look at type 1 and type 2. Type 1 topoisomerases increases the ALK by excuse me. It increases the linking number in steps of 1. What does that mean? So, on the right hand side again we come to the same model. It has a linking number of 32, we had just talked about, it is the supercoiled DNA. The twists theoretically calculated are 36 and it has now 4 writhes. There are 4 times the DNA is rotating around its own axis. So, right handed helix, right interwound helix, so it is a -4. So, this is the twist number which is there. Now, what a type 1 topoisomerase does is that it introduces an extra twist. So, when it introduces an extra twist, the linking number increases by 33. So, if you can look at, the writhe remains the same as -4, but the linking number increases. So, but the linking number increases. So, type 1 topoisomerase introduces, if you see in the model of the DNA, in the backbone of the DNA, then what it is doing is that introducing a twist, and now to accommodate this twist, the writhe has to reduce. So, it increases the ALK by 1. So, it was 33 and 37 plus -4. So, to remain that, but still because it has increased the twist number, the writhe will have to decrease. So, finally, the linking number will be increased by from 32 to 33, but the twist increased will be accommodated by the reduction of a writhe. So, initially there were 4 writhes, 4 times it was rotating on its own axis, and on the other side you can see now it is rotating only 3 times. So, the writhes decrease, the twists increase, and the linking number increases by the step of 1. So, every time the type 1 topoisomerase act on the DNA molecule, it increases the linking number by 1. Shortly we will talk about a little detail about the mechanism also, but that is how it works. If we look at the type 2 topoisomerases, they change the linking number by steps of 2. So, they change the linking numbers. How do they do that? Then another important factor is that type 2 topoisomerases requires the energy. They requires ATP basically to do this. So, if the linking number was 33, now it has 32 to start with for a supercoiled DNA, now it has increased to 34. And how it has done that? By reducing the number of writhes. If you can see in the figure which is given below, there were initially 4 writhes which was there, and now it has reduced to the 2 writhes which is there. And that too, it changes the handedness of the turns. The topoisomerase 2 changes the DNA topology. So, that is how it has done it. So, if you can see, the linking number has risen from 32 to 34. It does not affect the twist. It does, what it does is basically reduce the number of writhes. So, the 4 turns which were there inside in the DNA double helix, in the DNA axis of the DNA molecule has now reduced to number 2. There is another enzyme which we talk about, is called as the DNA gyrase. DNA gyrase decreases, it is present in the prokaryotes, it decreases the linking number by 2. So, basically what it does it, that in the relaxed DNA, initially we were talking about the supercoiling changing to the relaxed state. Now, we are talking about the relaxed state changing to the supercoiling state. So, there is an enzyme which is present in prokaryotes which can decrease the linking number by 2. So, to start with, the linking number here is 36, and if you can see, then this DNA gyrase will introduce a writhe, and by introduce the writhe, it has to accommodate by doing the twist. So, it will introduce a writhe, it again requires ATP energy, then by introducing a writhe, it will require the ATP energy to be there, and it will be accommodated by the presence of the twists. So, DNA gyrase decreases the linking number by 2 steps. Now, this is a comparison again for class 1 and class 2 topoisomerase, just in a tabulated form. So, you can understand, and we will do a little more mechanism, that class 1 topoisomerase cleaves the one strand, whereas and that cleavage is called as the cleavage or cut. It basically does it at one strand, but class 2 topoisomerases create a break. It breaks both the strands. So, delta L is changed by the steps of 1, and delta L in case of class 2 topoisomerase changed by the steps of 2. So, the mechanism we will describe in detail a little bit. ATP requirement, the class 1 topoisomerase do not require ATP, whereas class 2 topoisomerase does require energy. Examples are in E. coli, the topo 1, topo 3, they relaxes the negative supercoils as we just saw. And in class 2 topoisomerases, we have gyrase that introduces the negative supercoils, and another one which is called as topo 4 that decatenates the linked circles. If the two DNA molecules are linked, then it breaks both the strands, and then it will separate the two circles. In eukaryotic systems, there are class 2 topoisomerase 1, it relaxes both the positive and negative supercoils. So, from supercoiling to a relaxed state, and topoisomerase 3 which relaxes the negative supercoils. In class 2 topoisomerases, they have topoisomerase 2 that again relaxes both positive and negative, and topoisomerase 4 that relaxes the negative supercoils. So, topoisomerase 1 and 2, which are primarily called as, are reducing the negative and the positive supercoils, and DNA gyrase is increasing the supercoils. So, a relaxed state is converting into a supercoiled state by DNA gyrase, and topoisomerases are maintaining them to become the relaxed state. So, that is how they create a balance between whether the DNA has to occur in a relaxed state or no over supercoiling occurs, as it is called as. So, the topoisomerases play a role. So, we will look a little deeper into the mechanism of action of these type 1 topoisomerases, because that will make you appreciate that how the mechanism works, and will put to use the understanding of the supercoiling of the DNA which we just talked about, and will reinforce that, will re-emphasize that understanding. If you look at it, the first step of the cleavage will be the strands of the DNA separate. So, you can see that there is a supercoiled DNA, there is a supercoiling DNA, and when the supercoiling opens up, then there the two strands also separate. So, the first thing will be the strands of the DNA separate. This is very important point, because a type 1 topoisomerase can only act on a single strand DNA molecule. It cannot act on a double stranded DNA molecule. The two strands have to be separated for the type 1 topoisomerase to bind to it. So, and this separation can occur, as I told you, when there is an over supercoiling. Then over supercoiling, and when it causes relaxing, then by itself it can relax and can generate the separate two strands. So, the strands of the DNA are separate. The type 1 topoisomerase will come and will bind at one of the strands. Particularly, it will bind at the cleavage. The cleavage site will be the active site tyrosine, which is basically bound to it. So, the type 1 topoisomerase will bind to the DNA molecule by placing the active site tyrosine in which is present inside the catalytic cavity just near to the DNA strand. So, it will do that. The next thing will be that it will take up a proton from a very basic amino acid residue which is lying nearby to it, and create a oxyanion. So, this oxyanion which is created on the tyrosine will create a kind of an intermediate. So, type 1 topoisomerase binds one of the strands, and if you can see now, this oxyanion will act on the phosphate, nearest phosphate bond. So, the tyrosine residue was there. This tyrosine residue in the catalytic activity will act on the phosphodiester bond. If you remember that nucleotides, the sugar, phosphate, they are attached. So, they will act on the phosphate bond, and to attack on the phosphate bond, the oxygen, which is basically hydroxyl molecule, has to be oxyanion, and this becomes oxyanion by taking a proton from a nearby basic amino acid residue. So, now it will attack this phosphate bond, and then this phosphate bond will break. So, it will form an intermediate which is a 3' phosphotyrosyl enzyme intermediate, and then there will be a 5' hydroxyl group. So, the 5' hydroxyl group, which is a phosphodiester bond, basically has been broken. That is what it has to do. It has to cleave the DNA. So, if you remember, then the backbone of the DNA is a sugar phosphate backbone, and how you can cut this DNA or how you can create a nick in this DNA is by nicking this phosphodiester bond. So, it has broken up this phosphodiester bond. Now, it will form a DNA 3' phosphotyrosyl enzyme intermediate, and the 5' hydroxyl group is formed by taking up the proton from an acidic amino acid residue which is lying in the near cavity. So, it will take that proton and it will become the 5' hydroxyl group. So, it has created the type 1 topoisomerase came, bound to one of the strand, and now has created the nick. And then after creating this nick, a conformational change will occur in the type 1 topoisomerase, and it will increase this gap. So, the two strands are broken, and now these two broken gaps are held apart. So, that the conformational change, the enzyme undergoes a conformational change, opening a gap in the DNA molecule. The next step will be then the topoisomerase enzyme will take this uncut strand, and then it passes through the gap, and then the enzyme closes. So, it has created a twist basically in the DNA molecule, where there was a gap which was present. Now, it has taken from one side and passed it over to the another strand, and where the two strands pass over one another, that is the turn, that is our linking, that is how a twist is created. So, it has taken the two strands which were separated, taken one strand and passed over to the another strand. So, uncut strand passes through the gap, and then the enzyme closes. So, enzyme closure is a reversal of the reaction which happened. So, it will be the same reversal. The 5' hydroxyl group will give back the proton to the amino acid residue, and then the phosphate bond will be formed with the sugar of the next one, and then tyrosine residue will again give back the proton to the basic amino acid residue, and the topoisomerase, this ligates then nick the strand. So, whatever cleavage mechanism was there, exact reversal will happen, and the strand will be ligated together again. So, the topoisomerase then releases to find another thing. So, at one step it has introduced one twist. So, if you can see here, there was a gap which was there. It was a gap which was basically there. There was a single strand, two strands were separated, and at the end what has happened? There has been a twist which has been introduced. So, the topoisomerase has introduced a nick. So, to start with, the linking number was something. If we look at this, what is the change in the linking number now? Now, the final linking number, the twist has been introduced. So, initially there were 2 twists, and now there are 3 twists. So, linking number is increasing by the number of 1. So, topoisomerase 1 acts in this manner, and therefore, obviously if it is introducing a twist, then it is changing the supercoiling nature of the DNA. So, from it is relaxing the supercoiling DNA, or it is changing the positive and the negative supercoils. So, this is how basically again a broader thing, how topoisomerase 1 works on a single strand, and now we will look at how topoisomerase 2 works. So, in the lower figure if you see, then topoisomerase 2, the break is occurring in both the strands. So, it cuts both the strands, the phosphodiester bond on both the strands, and then passes one over the another, and that is how it works. So, the next figure talks about the mechanism of action. I will not go into too much of detail of this thing, but just to have an overview so that you can appreciate the two different kind of topoisomerases and how they work. If you look at it, it starts from the left hand side, that is the enzyme, and the enzyme has two conformational places. One is the ATPase domain, because as I told you, the topoisomerase 2 requires energy, it requires ATP. So, that is the ATPase domain where the ATP will bind and will be cleaved to provide energy, and then there is a DNA cleavage core domains. The mechanism which I am talking about, even for type 1 topoisomerase or type 2 topoisomerase, is not a general mechanism for all the topoisomerases. Many of the topoisomerases work in this manner, but there are many other topoisomerases which work in different manners also. But these are the generalized which we are talking about here. So, there is an ATPase domain and there is a DNA cleavage core domain. So, the DNA will bind, it will bind to one strand of the DNA, then the ATP will bind to the ATPase. When ATP will bind to the ATPase and then ATP will be utilized, then the second strand will be bound to this. The first strand will be cleaved, the second strand, the strand passage, the fourth number. So, the second strands will pass through that catalytic cavity to the other sides through the cut of the first one. So, the first strand will be cut, the first molecule will be cut, DNA molecule, the second part of the DNA molecule, just excuse me, this is not the strands I am talking about here, this is the two different DNA molecules which are, so entire DNA molecules passes on to one another. So, topoisomerase II is working on the double stranded DNA molecule, it does not require single strands. So, it gets hold of one part of the DNA molecule and brings the other part of the DNA molecule, then cuts one part of the DNA molecule, passes the other strand passes the other DNA molecule towards that passage and then religates it. So, that is how it is creating a supercoiling, it is relaxing the supercoiling, or that is how it is introducing this thing, and therefore, it requires the energy for that. So, as you can see, there are double stranded molecules which are attached and double stranded, so it does not require the single stranded DNA molecules. Now, if we look, so this was all about the just to look at like how DNA is present inside the cell as far as the supercoiling aspect is concerned. Then we go in for a higher level of organization, that with this supercoiling, then how these strands are arranged, which we will be talking about in eukaryotic genome organization. But then there are certain aspects to it, like how can we distinguish between these two DNA topoisomers. If you isolate a plasmid DNA, for example, it is a circular DNA molecule, and how do you visualize plasmid DNA? You run it on a gel electrophoresis. So, when the gel electrophoresis, DNA is a negative molecule, it will run towards the positive side. So, when it runs towards the positive side, it gets separated based upon the size. Gel is a porous structure. When the DNA molecule runs through it, then the smaller molecules or less molecular weight molecules tends to run faster, the bigger molecules tends to run later. So, they will come towards the well and the smaller ones will run through that. So, the DNA topoisomers which we have talked about, because they are the same sequence, the same length DNA. So, the same length DNA, same sequence DNA, but when you isolate and when you run it on a gel, then you can separate these topoisomers, because when a relaxed DNA is supercoiled, then it occupies less space. So, it is a smaller molecule. So, supercoiled DNA is smaller than a less supercoiled DNA molecule. So, supercoiled DNA molecule will run far ahead in the gel electrophoresis. However, the relaxed DNA molecule will come a little later. So, even if the weight is same, the base pair is same, the length is same and everything is same, they can be separated in this manner. That how they can be done. I don't know what happens. So, if you can see, then the topoisomerases can be separated by gel electrophoresis. The supercoiled runs the farther away from the where it is loaded, and the relaxed one comes at the back. So, that is how the DNA topoisomers can be separated. Now, to visualize, I just want to talk about this factor here, because we are talking about the supercoiling. Then to visualize a DNA on a gel, we need dyes. DNA is a transparent molecule when you isolate it. If you want to visualize it, you need to put some color to it, and this color is basically a most common color which is used, a dye which is used is called as the ethidium bromide dye. So, you stain it with an ethidium bromide dye, and when you visualize it under UV ultraviolet rays, then it fluoresces, it gives a color, and that is how you can visualize. Otherwise, in a with normal eye outside, you cannot, in a normal light, you cannot identify the DNA molecules which you have run on a gel. So, this ethidium ions, how does it work? It intercalates between the two strands of DNA. So, the ethidium ions cause the DNA to unwind. So, that is also one important aspect, that these intercalators means which is introducing or which is sticking inside the two strands of the DNA. So, as you can see, that is the DNA molecule, and those red lines are the ethidium bromide. So, when you add the ethidium bromide to DNA molecule, then it just goes and intercalates between the two strands in the DNA helices, and this intercalation reduces the twists, because it opens it up, it reduces the twists, and therefore increases the writhes in a closed circular molecules. And for some of the things, it does affect the restriction digestion of the DNA also. And that is why I just introduce it, because down the lane when you talk about the restriction digestion, then if you have treated your DNA with the certain dyes, then the digestion is affected. So the digestion, why it is affected? Because it reduces, because it is playing with the topology of the DNA. It is basically supercoiling of the DNA, it is changing. So ethidium. So you can see here, if you can that there is a writhe which is 0 in a relaxed state, and if you add the ethidium ions, then it can go from a negative supercoil to a relaxed state, and if you still add more ethidium ions, then it can go to a positive supercoil also. So these intercalators can introduce or relax a negative supercoil. So just like Topoisomerases was working, but it's not exactly like that, but these are because they intercalate between the two strands of DNA, so they relaxes a negative supercoil, and if more of them are introduced, they might twist it into the other direction and can introduce the positive supercoils also. So that's all about the DNA topology which I wanted to talk about. The next aspect which I want to talk about today is the DNA melting or DNA denaturation. When two strands of DNA, the two strands of the DNA have to be melted or denatured, which we are talking about, can occur due to certain factors. So that refers to the melting of the double-stranded DNA to generate two single strands, and this is the property of DNA which is utilized for all the processes that DNA undergoes. For replication, DNA has to become a single strand. For transcription, DNA has to become a single strand. And we also have to understand that what are the stages in which the DNA can become a single strand. So DNA melting refers to the melting of the double-stranded DNA to generate two single strand. It involves breaking of hydrogen bonds between the bases in the duplexes, and it also involves disruption of the base stacking, that is native and denatured. If you can see, then there are certain factors which are there. There is heat, there is non-aqueous denaturing solvents, there is low or high pH, and there are lower ionic strengths. Whereas the clock tells me that the time is running out, and I think I will be able to continue this in my next exercise, the DNA melting as well as the organelle DNA. So,
thank you, Dr. Charu Dogra, for a very interesting lecture. I hope I have never heard anyone explain linking, twist and topology so easily. I'm sure the students would like to read more about it, would like to solve more numericals about it. I had a few questions, but as time is running out, I just want to ask you that does B, A and Z DNA occur normally, and can we find these topoisomers occur in the normal DNA when there is replication and transcription occurring? So we might be having the occurrence of several topoisomers even when the DNA is occurring normally and is functioning.
Yes, I'll take your first part of your question first, that normally the DNA occurs in the B form of DNA. The A form of DNA and Z form of DNA are because of the variations in the water content of the DNA. So there are studies which are now showing up that there might be a presence of these forms of DNA naturally also. But when they were found out, the A form of DNA and Z form of DNA, they were in the tube. They were not existing in the cell. Normally the DNA exists in the B form of DNA. The second part is when you talk about the topoisomers. So of course topoisomers are present inside the cell, because that's how the cell in the cell the DNA might exist. It can be in the supercoiled form, but it can be in the relaxed. When the replication has to occur, it has to come to the relaxed form, some kind of relaxation. So entire DNA or the entire genome, we cannot define that whether it will be in the relaxed form or the supercoiled form, but yes, the parts of DNA we can talk about that some parts of DNA at some stage under some conditions are in the relaxed form, and some parts of DNA are under the supercoiled form.
Thank you, Dr. Charu Dogra, for explaining these queries to me. We will follow up with the DNA denaturation and genomic and subgenomic DNAs in our coming lectures. Thank you, Dr. Charu Dogra. Thank you, viewers. You can catch this lecture on the YouTube channel, and please do send in your queries and any suggestions you have at the email addresses given below. Thank you.